Alright, so this is my department thesis proposal, which is titled An Investigation into Novel Techniques for the Genetic Manipulation of Aedes aegypti. So as a brief outline, I'll go over Aedes aegypti as a vector, which is a species of mosquitoes. I'll then go over both of my objectives, give a brief summary, and lastly, show my acknowledgement. So, Aedes aegypti is an important disease vector. It's known to vector such diseases as dengue, chikungunya, and yellow fever, among others. But those are the three big ones, dengue being probably the biggest one. As you can see in this map, which is a little bit older, but it's gotten worse since then. There's a large area of possible, or where you could get infected with this disease. According to the World Health Organization, there's greater than 2 billion people are at risk, and 50 to 100 million people get infected every year with dengue. I think it's between 10 and 20,000 people a year die, but the rest suffer from the disease. It's known as break bone fever, so it makes you feel like your bone's breaking. So it's pretty nasty stuff, and it's pretty widespread, and there's really no treatment for it. So kind of a nasty disease. Here you can see the Caribbeans, which recently, just kind of to show that this vector is out of control, the diseases that it's carrying anywhere they can be. Chikungunya, one of the diseases which Aedes aegypti vectors, just recently popped up in the Caribbean. Now, it's normally native to Southeast Asia, I believe. Within the past couple of years, it's popped up in Europe for the first time, and now, within the past couple months, I believe it was December, it showed up in the Caribbean. So these guys are spreading these diseases everywhere, and they're bad guys. So my first objective has to do with the non-homologous end-joining pathway, which is a cellular repair pathway. So as you can kind of see in this image up at the top, you have the your DNA is broken in half, or any kind of action which causes damage to your DNA, which causes it to effectively break, is bad. So your cell has evolved certain pathways to deal with that kind of breakage. One of which is the non-homologous end-joining pathway, as you can see on the left. So the non-homologous end-joining pathway is just a cellular repair pathway which repairs broken DNA. In the process, it creates either deletions or insertions. So it repairs it relatively quick. It's the go-to method, but you have small insertions or deletions of DNA. So it changes the code effectively. On the other hand, as you can see on the right, an alternative repair pathway is the HDR, or the homology-directed repair pathway, which, unlike the non-homologous end-joining pathway, leads to an accurate repair of the DNA which was broken. The only caveat being that it requires a donor template. So there's certain times in the cell life cycle in which homology-directed repair cannot occur because there is no donor template, which is why you have the non-homologous end-joining pathway. So why exactly are these repair pathways important? So recently, gene editing tools or genome editing tools have utilized these repair pathways specifically by causing double-stranded DNA breaks. So in our lab, for example, we were able to knock out the pigment in the mosquito's eye utilizing the non-homologous end-joining pathway to cause a double-stranded break in that protein, which created usually deletions, but sometimes insertions, which effectively knocked out or changed the shape or knocked out the protein itself. So the non-homologous end-joining pathway has been utilized before. What we'd like to do is to utilize the homology-directed repair pathway, because you can use the homology-directed repair pathway by introducing a donor template that you've designed to insert marker genes or whatever you want to insert. So it'd be more beneficial to utilize the homology-directed repair pathway. Now, previous studies have shown that if you knock out components of the non-homologous end-joining pathway, the pathway on the left, then there seems to be an increase in the utilization of the homology-directed repair pathway, which is what we would like to do because the rates of homology-directed repair right now, at least what we're seeing in experiments, is extremely low. So, if we were able to create a tool which allowed us to monitor the activity of the non-homologous end-joining pathway, it would be extremely beneficial to this whole effort. Which brings me to my first objective, which is the creation of a reporter plasmid with the ability to assess the activity of the non-homologous end-joining pathway in AG2 cells, embryos, and mutant embryos. My hypothesis being the alteration of essential components, the non-homologous end-joining pathway, by knockdown or knockout, will suppress the activity of the non-homologous end-joining pathway. So the first step is the creation of the reporter itself. So here's an overall picture of what exactly the reporter plasmid looks like and how it's going to work. So if you look here, up here is the actual reporter plasmid itself, which has a frameshift mutation here at a specific site, and this mutation is due to the insertion of a cleavage, cleavage domain, which is specific to the IANI1 endonuclease. Now these poly-UV sections up here are just promoter regions of these two pieces of DNA, the top one and the bottom one, which are endogenous to Aedes aegypti. So pretty much what that means is these promoters allow for this firefly protein and this IANI1 endonuclease to be produced in the cell. So you have both of these guys present if your plasmid, so this IANI1 endonuclease comes in and cuts this plasmid here, and if this plasmid is repaired by the homology-directed repair pathway, you should see no increase in firefly luminescence, versus if it's repaired by the non-homologous end-joining pathway, you should see an increase in luminescence. And I forgot to mention, but firefly, the protein, is an enzyme which is utilized in the firefly, American firefly. So it emits a light, effectively, and you can use the emission of this luminescence to quantify relative rates and populations of your plasmid. So, how exactly does it work? So here we have the poly-UV promoter driving the firefly luciferase protein, which is a pretty standard tool we use in our lab. You insert the IANI1 cleavage domain at the specific site. This truncates the protein, effectively making it about a third of its normal size. So it prematurely cuts the protein down in size, which reduces luminescence. If the IANI1 endonuclease is present, it'll come in causing a double-stranded break, which, if repaired by the homology-directed repair pathway, leads to no significant change in luminescence. But if it's repaired by the non-homologous end-joining pathway, about a third of the time, due to the nature of genetic translation, you should see an increase in luminescence due to the firefly protein's restoration. And I think I'm about out of time. I can only do some 10-minute increments, so go ahead and start up with the next video.